Hello, everyone, and welcome to today's live broadcast, Post-Genetics with the new Axiom Human Genotyping SARS-CoV-2 Research Array. My name is Andrea Klein, and I'll be your moderator. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. This is the first topic in the new webinar series on predictive genomics for population and personalized health. For more information on our sponsor, please visit www.thermofisher.com. All right, let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you like at any time during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. Also, please notice you can share this webinar on your personal social media. Just click on the Social Sharing tab to let your friends and colleagues know about today's event. If you have any trouble seeing or hearing the presentation, you can click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. You can click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd now like to introduce our presenter, Shantanu Kaushikkar, Director of Product Marketing for Microarray Genotyping at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. For the Q&A session at the end of the presentation, Shantanu will be joined by Dr. Jeremy Golub, Director for Bioinformatics at Thermo Fisher Scientific, and design lead for the Axiom Human Genotyping SARS-CoV-2 Research Array. Shantanu, I'll now turn it over to you to begin your presentation.
Hello, and thank you for the introduction. And thank you for attending our presentation today on the new Axiom Human Genotyping SARS-CoV-2 Array. In the next hour or so, you'll hear from me about the use of this array for research in host genetics and development and vaccines and therapeutics in SARS-CoV-2 infections. The agenda for today will cover how Thermo Fisher is enabling research in SARS-CoV-2 impact during past outbreaks and our role in SARS-CoV-2 research efforts. We'll speak about the role of microarray as a whole genome scalable technology for rapid research. And then we'll dive into the design of the new Axiom array that reflects a paradigm shift in array design to address the threat from this virus. Lastly, we'll wrap up with a question and answer session. Thermo Fisher has a history of response in public health crisis. We've worked hard to make a difference during not just this SARS-CoV-2 outbreak, but also during Ebola, H1N1, SARS, and Zika too. While we are talking about our genetic analysis solutions for outbreak research today, Thermo Fisher also has a hand in PPE, other scientific research tools like those for protein and cellular analysis, in detection kits, and in vaccine production and clinical trial logistics. We offer a broad range of technologies for genetic analysis, including whole genome genotyping and whole transcriptome microarrays, next-gen sequencing, capillary electrophoresis for Sanger sequencing and fragment analysis, and of course, real-time PCR. Microarrays play a unique role in supporting SARS-CoV-2 research because of their capabilities in whole genome genotyping and in whole transcript expression analysis. Here at Thermo Fisher, we are committed to service science in the battle against SARS-CoV-2 infections. We have genetic analysis solutions for virus sequencing, pathogen detection, epidemiological surveillance, vaccine research and development, and host genetics and population genomic studies. Within host genetics and population genomic studies, our whole genome microarrays can help identify host genetic factors potentially associated with susceptibility, resistance, and more. Our portfolio of genotyping and gene expression arrays offer the capability to get a whole genome view. In vaccine research, the microarray platform can help identify and study the impact of infection and potential vaccines on immune response. Let's now discuss the role of microarrays in addressing the challenges posed to our world by the SARS-CoV-2 virus and how the Axiom genotyping platform can be used for research in host genetics, vaccine, and drug development. In this slide shows age, gender, and ethnicity have been reported as prognosticators of SARS-CoV-2 infection susceptibility and severity. But there are a range of other factors that can impact the severity and susceptibility to the disease. For example, the CDC released the results of their pathology and pathogenesis study last week that highlighted the role of pre-existing conditions such as diabetes, lung function, and cardiovascular disease in severity of SARS-CoV-2 infections. Other conditions that have been reported in various studies since the incidence of this virus that can impact severity have been hypertension and cancer, to name a few. For example, the NIH Director's blog recently mentions correlation between ABO blood group types and increased susceptibility to the disease. Vitamin D levels, blood groups, and body mass index have also been implicated in the severity of the disease. The various outcomes that have been reported in literature and in news articles have included respiratory distress, pneumonia, blood clotting, loss of smell, to name a few. The challenge for us then is to understand how to look at the interaction between the virus and the human body and understand the host viral response. The goal is to stratify population by risk and accelerate the development of vaccines and therapeutics to fight or even slow down the virus. There are multiple confounding factors when researching host viral response to SARS-CoV-2 infections. There is the dynamics and interplay between demographics, underlying conditions, and then the severity susceptibility to SARS-CoV-2. Susceptibility to the virus varies in populations, and here there are several processes involved in viral binding to the cellular membranes. The immune systems in each of us respond differently to the virus, and there are significant gaps in our knowledge in developing vaccines and therapeutic treatments. Genotyping and genetics can help address these challenges. To address these challenges, we need to screen genes in several thousand, if not hundreds of thousands of people. There is therefore a need for a low-cost, scalable, and fast technology, such as microarrays, that can overcome these challenges. The Axiom Human Genotyping SARS-CoV-2 Research Array was developed specifically for the purpose of research into host genetics. The array was designed with the help of key opinion leaders in this area. Several of the institutions and the individuals that helped us came from the institutions that helped contribute to the content that are listed here, and we would like to acknowledge their contributions and efforts. We would also like to acknowledge the role of the various consortia and the biobanks, such as those listed here on the left, including the UK Biobank, FinGen, Million Veteran Program, and the Spanish Biobank, that have continued to publish their research during this time. Some of the content that's included on the array is from the findings of these research studies. The new Axiom Human Genotyping SARS-CoV-2 Research Array includes all of the benefits of our modular approach to designing genotyping arrays, such as a large GWAS grid with multi-ethnic population coverage. But more specifically, this array includes 3 new modules with new and relevant content for studying susceptibility, severity, and immune response. The first module, called the SARS-CoV-2 Susceptibility Module, includes structural variants for viral entry points and pathways that are associated with immunity and inflammation. The second module includes discoveries and variants associated with chronic underlying conditions that are implicated in the severity of the disease. Also included in the module, in this module, are markers covering blood groups and blood phenotypes that have been implicated in the severity of the disease. The third and final module includes variants to cover regions associated with immune response and that are important in vaccine development and in pharmacogenomics for drug discovery and development. The module also includes new content on immunoglobulin heavy chains locus. This is something that was newly introduced for the purpose of immune response for SARS-CoV-2 infections. We will cover the modules one at a time in the next few slides. One of the most important aspects of this array is that it offers dense coverage of genes that are most relevant in SARS-CoV-2 infections. For example, the array offers fine mapping of ACE2 and the TMPRSS2 genes that are the viral entry points. There are two pictures shown over here on this slide that show the coverage on the Axiom array and then compares it with the global screening array, the GSA-V3. The two pictures show the markers that are designed on the array and then overlaid on the gene. As a comparison, the global screening array, for example, offers only 5 markers on the ACE2 gene. This is shown in the bottom picture, and you can see the trace for the global screening array. In comparison, the Axiom human genotyping array offers more than 750 markers for this gene, providing fine mapping capabilities. The reason for designing as many markers on the ACE2 and the TMPRSS2 gene is so that we can better understand the facilitation of the viral binding process with the cellular structures. The genes also serve as druggable targets, and fine mapping can help with research in both therapeutics and in vaccine development. The susceptibility research module also includes pathways that are valuable in genotyping and GWAS studies. This is because they allow the interpretation of variants with respect to the biological processes in which the affected genes and proteins are involved. Genotyping markers for pathways implicated in viral interactions can help conduct research into cellular mechanisms that regulate viral entry, infection, and immunity. These pathways also provide insight into vaccine research, drug-gene interactions in antiviral therapeutics. These pathways and structural variants were specifically selected because of their role in immune response, viral replication, inflammation, and immunity, including innate immunity. The pathways can help us decipher the underlying host genetic differences and individual responses to the SARS-CoV-2 virus. These are key to deciphering the SARS-CoV-2 susceptibility and severity to the SARS-CoV-2 infections. Some of the more important pathways that I'd like to mention over here include the NOT signaling pathway that's shown in the bottom left, the IFN pathway that's shown at the very bottom with more than 2,600 markers, the FOXO pathway with over 2,300 markers, and the NF-kappa beta signaling pathway that is involved in activating genes, immune response, and inflammation. The array has coverage for cytokine genes that are implicated in cytokine storm affecting outcome in infections. Another example is the interferon signaling pathway, which responds to the presence of cellular changes suggestive of viral infection. By inducing numerous genes involved in inflammation, signaling, and immunomodulation to protect against uncontrolled infection. They interfere with viral replication by slowing cell metabolism, interfering with membrane formation required for virus replication, and inducing secretion of cytokines that promote adaptive immunity. There is a paradigm shift, therefore, in how this array was designed to include the variants for these very specific metabolic signaling pathways. This slide covers the variants from the severity research module. It offers coverage of variants that are associated with underlying conditions and the traits that have been implicated in the severity of the disease. The content includes recommendations from key opinion leaders and from peer-reviewed publications. The blood module that has more than 2,000 variants, for example, provides the capability to look at the role of blood groups not only in susceptibility but also severity. This array offers the capability to make new associations while leveraging peer-reviewed content for the purpose of studying the severity in SARS-CoV-2 infections. The array covers diseases such as COPD, asthma, lung function, diabetes, hypertension, stroke, heart disease, and so forth. The third and final module offers coverage of variants that are important in drug discovery and development, in vaccine development, and in vaccine response. For pharmacogenomics, in drug discovery and development, the array offers more than 7,000 variants that offer high evidence and cover pseudogenes such as the CYP2D6. The array also offers variants for circulating proteins that are potential drug targets. Within the role of vaccine development, this array offers variants within the immunoglobulin heavy chain locus and microRNAs that are implicated in the development of vaccines. These more than 12,000 variants are novel and are designed on the array for the sole purpose of performing vaccine development and vaccine research. Finally, the last set of variants on this array includes variants from the HLA-KIR region that are typically involved in vaccine response. More than 13,000 variants offer coverage of the 11 major histocompatibility complex class 1 and class 2 loci within the HLA region. They offer 2- and 4-digit resolution and can be analyzed using the automated software for imputing HLA types from the genotype data. In summary, this slide shows the content that is essential to research host response, vaccines, and therapeutics. The Axiom Human Genotyping SARS-CoV-2 Research Array includes these 3 modules. The first module, the susceptibility module, includes structural variants from pathways, research into cellular mechanisms that regulate viral entry, infection, and immunity. It includes fine mapping of genes that are associated as viral entry points. The severity module includes disease-associated variants and can be used to leverage the genetic associations that have been implicated in underlying conditions. The array also includes a large GWAS grid to discover new disease associations. And then finally, the third module offers coverage of structural variants implicated in immune response and drug-gene interactions. This includes genetics from regions associated with immune response for vaccine research, such as the immunoglobulin heavy chain locus, and then it also includes markers from pharmacogenes for drug-gene interactions and in antiviral therapeutics. The Axiom Human Genotyping SARS-CoV-2 Research Array offers highly relevant content representing biological processes, genes, and underlying conditions for research into host genetics, vaccines, and therapeutics. The data from this array can be combined with data from other studies, such as those that have been run on the Axiom platform. Meta-analysis with existing studies will allow the community to develop approaches to identifying areas of the human genome that are involved in both susceptibility and severity. Collaborative approaches through meta-analysis and data sharing will generate insights and spark new ideas to help fight the SARS-CoV-2 virus. And with that, I'd like to thank everybody and move to the Q&A session. If you would like to contact us, then please email us at bioinformaticsservices@thermofisher.com, or please visit our webpage at the URL that is shown on this slide. Thank you.
Great, thank you, Shantanu, for your informative presentation. We will now start the live Q&A portion of the webinar and welcome Dr. Jeremy Golub to the studio. If you have a question you'd like to ask, you can do so now. Just click on that Ask a Question box located at the far left of your screen. We will answer as many of the questions as we have time for, and if we run out, you will have your question answered by the speaker using the contact information you provided at the time of registration. Okay, let's get started. Our first question is, given that type A blood has been found to be at significantly higher risk than type O blood, does this COVID array handle ABO locus on chromosome 9?
Hi, this is Jeremy. I can take that question. Yeah, in addition to the recent Ellinghaus paper, from which we picked up the key variants found in that research, the array offers excellent coverage of actually all 33 blood group systems in man, very much including ABO. In addition to the major subtypes, we can call many of the less common alleles of A and B as well as O. So that locus is quite well covered.
Excellent, great, thank you. Next question. On the slide discussing pathways, are you also covering Notch signaling pathways and genes covered in cytokine storm?
Yes, we specifically addressed Notch and the cytokines as well as the regulatory elements.
Okay, great. This one should be pretty straightforward. When is the array going to be available?
I'll take that, Andrea. So the array is available today, and the way we manufacture the array is once somebody places an order, it takes us about 4 weeks to make the array. So it should be available, if somebody places an order today, it should be available 4 weeks from now.
Excellent, great. Oh, go ahead.
No, go ahead. That's all right.
All right, great, excellent. Okay, another one. What markers associated with acute respiratory distress syndrome and stroke does the array cover?
So we have on the array a large number of variants associated with lung function relevant to ARDS. In addition, of course, we have the cytokines and interferons and the regulatory pathways affecting various aspects of acute respiratory distress. For stroke, we have many of the published GWAS hits relating to stroke, as well as good coverage of the coagulation pathway, since the connection to stroke here seems to be primarily unusual blood clotting or unusual degree of blood clotting, especially in younger patients. We took care to cover that pathway.
Great, great. Okay, next question. There was a recent publication from MVP that shared variants implicated in diabetes across multi-ethnic populations. Have you included those variants on the array?
I'll take that, Andrea. Thanks for the question. Yes, so the CDC recently identified diabetes as one of the underlying conditions that affects the severity of SARS-CoV-2 infections. We did include variants from this paper that was published in Nature in June. The author's first name is Vujokovic. The Millions Veteran Program cohort genotyped on the Axiom platform was one of the many used in this analysis. And this paper basically identified 318 new risk loci for type 2 diabetes and related vascular outcomes. And this was among 1.4 million participants through a multi-ancestry meta-analysis. So these are included in the array, in addition to variants from, of course, the NHGRI, GWAS databases and other peer-reviewed publications.
Okay, great. I'll end on this next question, but just want to remind our audience that the questions we were unable to answer today and those that come in during the on-demand period will be answered by Shantanu or Jeremy using the email address you provided at the time of registration. So our last question then is, does this array cover multi-ethnic populations? And if so, can you share more information on what populations are covered with this array?
I'll take that as well, Andrea. So the array covers the 5 ancestral populations. This is basically the European, admixed American, African, South Asian, and East Asian populations, and so covers the 26 populations from 1,000 genomes. And then in addition, there are various pathways, often include markers that cover the multi-ethnic populations. So hopefully that gives information on the populations. Just want to make an emphasis that this also covers admixed Americans, such as Latino, African populations, so very suitable for use around the globe.
Excellent, great. Well, thank you for that. And Shantanu, do you have any final comments you'd like to make before we close?
No, I think just a general comment. We designed this array, SARS-CoV-2 array, for specific reasons. I think if you consider other general purpose global arrays that are available out there, then typically there are 2 challenges. A more general purpose array is often one where genes are selected based on the etiology of other diseases. And so what happens is it carries the risk of losing focus and missing regions that are implicated in SARS-CoV-2. And further, in a general purpose array that we believe there's a lack of statistical powers, so you end up typing SNPs that are not of specific interest to infectious disease, and so you run the risk of false positive inflations. The genes, on the other hand, the genes on the Axiom human genotyping SARS-CoV-2 array were selected with the very idea of their involvement in susceptibility and severity of the COVID-19. So it makes it really ideal for research in the ethiopathology of COVID-19. The last point is we have a promotion running on this array until September. So if there is anyone that's interested, please reach out to us. And we'll have one of our reps reach out to you and provide information.
Excellent, great, thank you. I want to thank the audience for joining us today and for submitting their questions. Again, any not addressed today will be answered using your contact information provided. So I'll thank again Shantanu for his time. We want to also thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, so LabRoots will alert you by email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. And with that, we will sign off. I hope everyone has a great day.